Okay, so good afternoon, everyone, and thanks for taking your time to come to my talk. And as Vivian mentioned, I'm a new assistant professor. Actually, my colleague told me, say I'm a baby professor in the Department of Neuroscience and Physiology at SUNY Upstate Medical University. And I start my own lab since last September, so it's like pretty new. So today I want to share my postdoc study, which done in June Wen's lab at UCSD, and we want to address how we prioritize behavior, and most specifically, how I prioritize behavior at the right time. I share my talk. I want to show you a video with you and first, and we can go through my my talk.
Hello.
What are you doing?
I'm just contemplating Buridan's donkey.
I understand. I'll leave you be.
What, you're familiar with the reference?
Of course. Jean Buridan proposed a philosophical thesis stating that a hungry donkey placed between two equidistant bales of hay would be paralyzed by indecision and would starve to death.
Exactly.
I wouldn't want you to starve to death, so here's an eggplant.
Okay, so as Amy mentioned in this video, inability to prioritize behavior lead to a catastrophic outcome best displayed by Buridan donkeys. A failure to decide whether to eat or to drink proven fatal to the Buridan donkeys. However, in the wild, we don't see a lot of Buridan donkey, right? This suggests that there are neuronal mechanisms to prevent this such catastrophic indecision in animals. And a famous neuroethologist, Conor Lawrence, studied indistinct behavior in animals. In his seminal work on aggression, he proposed the great empowerment of the instincts as a model to explain the interaction between the different behavioral motivation systems. However, in my mind, the great empowerment of the instinct hypothesis, since a winner tests all models in which all drive compete and only one winner emerges. But when I read this, I have to say I'm quite surprised by the idea of the complete system of the interactions, because it would be very costly to wire up all the motivational system in the neural systems. By contrast, the psychological study in humans, Abraham Maslow proposed that the motivational system is organized in a hierarchical manners. So when all the physiological needs, such as eating, drinking, has been met, and safety needs become the priority, and when the safety and physiological need has been satisfied, then the needs for the belonging and love will be prioritized. Maslow's hierarchy of the needs is widely accepted, despite there is a little experimental data to support it. So here is an example of the human study. In 1944, the health men's volunteers were recruited for the Minnesota starvation experiment. So in this experiment, their goal was to find the best way to help the war victims to recover from the starvation. And the figure shown here is a comparison of the frequency of the time thinking about food and thinking about sex. And as expected, many of the starved volunteers report spending most of the time to thinking about food. In contrast, they spend much less time thinking about sex. And this result suggests that the sexual motivation is suppressed by the starvation. And this is a striking example that supports the Maslow theory. The lower level of the need must be met before the higher level of the need can emerge. So in the recent years, Maslow's hierarchy of the needs has been adapted for study animal behaviors. Specifically, in animals, 3 layers of this hierarchy: physiological need, safety, and social need has been observed. Because we cannot define the cell extent and cell acutization in the mammals. However, our understanding of the biological basis of establishing such hierarchical organization is limited. Specifically, how animals would choose to display one particular behavior when the low level of the need has been satisfied is still unknown. So now I'm going to share our exciting finding on how priority is set and reset between the feeding and mating behavior in Drosophila. And in order to address this question, I use a Drosophila as an animal model to test the hypothesis of the Maslow's hierarchy. Low level of the need has been satisfied before the social behavior emerge. So we designed a behavior assay to test whether the fly prioritize feeding physiological need over courtship social behaviors. And in these assays, a male fly is given access to food and virgin female simultaneously. And then I recording feeding and courtship behavior for 30 minutes. And here, the feeding and courtship event are represented by blue and orange bar in the raster plot. So as you can see, 24-hour star males engaging in the feeding about 5 minutes before starting to court. And the video showing here, you can see the star male feed on the food. The fly is eating. And then they will transition from feeding to courting. You see, right now, the flies perform courtship behaviors. Okay, and by contrast, the sadist males call to female right away. So therefore, our observations are supporting Maslow theory. The lower needs, such as feeding, must be met before the social behavior are expressed. So now with this assay, I want to investigate how the animals transition from one behavior to another when low level of the need has been satisfied. So specifically, I want to address 2 questions in my study. First, what the signal molecule represent the lower level of the need has been made. And second, what is the underlying neural mechanism that regulates the switching from feeding to courtship in Drosophila males? So since this behavior switch occur at a short time period, it's about 5 minutes. So we hypothesize that the switch is mediated by the signaling molecule that represent the injection of the food. And to identify the signal molecule and neural substrate for the switching from feeding to courtship, we first look at whether the specific nutrient of the food induce this behavior switch. So in vertebrate animals, GnRH release respond to the minute-to-minute changes of the availability of metabolic fuels. So there are a lot of evidence supporting the notion that metabolic hormone regulate sexual motivation by modulating GnRH neuron in the hypothalamus. However, food is composed of different nutrients. It's unclear what nutrient through what metabolic hormone can regulate the behavior switch from feeding to courtship. So therefore, we first test whether the behavior transition from feeding to courtship is mediated by the specific nutrients. In the natural environment, rotten fruit are the good source for the Drosophila. and the yeast growing on the rotten fruit are the high quality of food source. So therefore, carbohydrates and protein are two important macronutrients for the Drosophila. So we decide to repeat the behavior experiment with either a 20 amino acid mixture containing food or isocaloric concentration of the sucrose. And surprisingly, the fry that consume sucrose do not transition from feeding to courtship. By contrast, male fry that consume amino acid transition from feeding to courtship after 5 minutes. So this result suggests that the amino acid promote the transition from feeding to courtship. Furthermore, this result argue that it's specific nutrient rather than the energy that regulate the transition from feeding to courtship. So we then hypothesize that the amino acid trigger the release of the signal molecule that induce the behavior switch. So as the behavior transition take around 5 minutes, as I mentioned before, and also require the amino acid consumption. So we hypothesize a metabolic hormone from the gut responding to the amino acid, but not sucrose serve as a signaling molecule from the behavior transition. And Park et al. Shows that there's one type of anterior endocrine cell in the gut called Dh31, which release Dh31. And this Dh31 positive anterior endocrine cell elevate the calcium level after amino acid consumption. So therefore, we use Drosophila with Dh31 knockdown in the gut to examine whether the Dh31 is required for the effect of amino acid on the behavior transition. So as expected, the control male switch from the feeding to courtship after amino acid consumption. By contrast, the Dh31 knockdown in the gut cause male to continue to feeding without initiating courtship, shown here. And this result suggests that the Dh31 in the gut mediates the behavior transition from feeding to courtship. And to test the time scale of the specificity of the Dh31 positive anterior endocrine cell response to nutrients, and we decide to perform the ex vivo calcium image of the fry gut. And we express the GCAM, the calcium indicator, in the gut Dh31 positive cell, and then we dissect the fry gut out and flatten the region where the Dh31 positive cell are expressed. And then we mount the fry gut in the perfusion chamber and examine the calcium response with the 1-photon imaging. So as shown in this video, we found that this Dh31 positive cell indeed respond to amino acid, but not sucrose and monosaccharides. So furthermore, we found a high concentration of the amino acid activates more Dh31 cells. And furthermore, its response increased with the concentration of the amino acids. So suggesting that Dh31 positive cell in the gut respond to amino acid in a dose-dependent manners. And now we know that Dh31, this neuropeptide released from the gut anterior endocrine cell, probably work as a hormone. So if Dh31 act as a hormone, we would predict the Dh31 level in the hemolymph would elevate after amino acid consumption. And hemolymph is the fry equivalent of the blood. So therefore, we use Western blot to measure the Dh31 level in the hemolymph. And the hemolymph of the wild type fry shows a significant increase in the Dh31 level after amino acid consumption compared to sucrose. By contrast, in the control experiment, Dh31 was not detected in the Dh31 mutant fry, as well as in the fry with genetic knockdown of Dh31. So collectively, this result indicate the injection of the amino acid increase the Dh31 level in the hemolymph. And next, we address whether an elevated Dh31 level in the hemolymph is sufficient for the fly to prioritize courtship over feeding. So we express CS crimson, a ligate ion channel in the Dh31 positive cell, and optogenetically activated cell for 5 minutes before asking the fly to make a choice between feeding and courtship. So the result shows here, optogenetic activation of Dh31 cell, start male initiate the courtship and ignore the food, while the control animal engage in the feeding before switch to the courtship. So this result suggests that the Dh31 from the gut is sufficient to suppress feeding motivation and promote courtship. So before I move to discuss how Dh31 from the gut act as a brain neuron to orchestrate this transition, and let me summarize the result I show you so far. First, the hunger fly prioritize feeding over courtship, and upon protein consumption, the male fly transition from feeding to courtship. And Dh31 is a protein satiety hormone that terminates the feeding, and Dh31 also can promote male courtship behavior. And the release of the Dh31 from the gut orchestrate the transition from feeding to courtship. And with that, this leads us to ask, what is the target of the gut Dh31 neuropeptides? So we start to look for the Dh31 receptor neuron in the brain, and luckily, the Porteous lab previously identified one type of the neuron called coexoning neurons in the fly brain are Dh31 positive, Dh31 receptor positive. And we also independently validate these coexoning positive neurons are also Dh31 receptor positive with antibody staining experiment. And coexoning is an analog of mammalian GnRH that promote reproductive behavior. So we then hypothesize that knockdown of Dh31 receptor in the brain coexoning neuron should abolish the switch from feeding to courtship. So when we knock down the Dh31 receptor in the coexoning receptor neuron in the brain, the fly barely engage in a courtship following the protein consumption shown here. And this result suggests that coexoning neuron in the brain is a target of the gut Dh31 for the behavior transition from feeding to courtship. So this result gives us an opportunity to test the nature of this gut-to-brain communication between the Dh31 positive anterior endocrine cell and coexoning neuron in the brain. So in order to test if Dh31 acts as a hormone, it is important to maintain the integrity of the circulatory system in the fly. And as we know, in traditional calcium imaging, we need to open the cuticle to visualize the neuron in the brain. However, unlike mammals, the fly have an open circulatory system. Removing the cuticle will perturb the circulation inside the body. And in order to investigate whether the brain Dh31 receptor positive neuron responds to circulatory Dh31, we need to image the fly brain with an intact circulatory system. In the other words, we need to use undissected fly to prevent the circulatory system for the functional imaging experiment. And our published paper in 2017 shows that three-photon microscopy provides a high-resolution transcuticle image in the fly brain. So here is a movie represent the transcuticle image of the fly brain by three-photon microscopy. The red is a three-photon signal. The green is a third harmonic signal indicating the non-neuron tissues. And using three-photon microscopy, we can clearly see the mushroom body structure through the cuticle without dissection. So here is a cartoon illustrated where is a mushroom body structure and where is it located. And here's a real image by the three-photon images. And you can clearly see the mushroom body structure right here without dissecting the fries. So with three-photon microscopy, we can image the chorizonin in the intake fries. So here is the chorizonin neurons in green, and this chorizonin express GCAM7, the calcium indicator. So to test whether the circulatory Dh31 can activate this chorizonin neuron in the brain, we first step inject the Dh31 synthetic peptide into the fry heart and measure the functional response in the brain chorizonin neurons. So here is a response of the chorizonin neuron to Dh31 injection. And this result suggests that the brain's chorizonin neuron respond to circulatory Dh31 neuropeptides. And similarly, when we optogenetic activation of the Dh31 gut cell was able to excite the chorizonin in the brain with a few minutes delays. And this time delay is consistent with the kinetics of the behavior switching from feeding to courtship. And also this response latency is variable with longer latency when the light intensity is lower and shortened latency when the light intensity is higher. So this response latency of 5 to 15 minutes from the Dh31 activation in the gut to excite the Dh31 receptor neuron in the brain, supporting the notion that the Dh31 indeed reach the brain through the circulatory system. And furthermore, if this gut-derived Dh31 reach the brain indeed through the circulatory system rather than a fast synaptic connection between the gut and brain, we hypothesize that stopping the circulation by cardiac arrest should increase the response latency of the chorizonin neuron in the brain. So here's a result. A brief exposure to CO2 causes a cardiac arrest, and heartbeat frequency are returned to the normal in about 5 minutes. And we observe a delay of about 7 minutes in the response latency of the chorizonin neuron when optogenetic activation of the Dh31 gut cells. So this time delay of the response latency is correlated with the period of time for recovery from the cardiac arrest. So supporting the Dh31 reach the brain through the circulatory system rather than a fast synaptic connection between the gut and the brain. So however, unlike the Dh31 knockdown in the gut, the fry with Dh31 receptor knockdown in the chorizonin neuron do not increase the duration of the feeding. And to further validate the Dh31 receptor knockdown in the chorizonin neurons do not have any impact on the feeding behavior, we decide to simply measure the total amount of protein intake by the feeding assays. So consistent with our previous observation from the behavior switching assay, we found that the Dh31 knockdown in the gut consume more protein containing food than the control. By contrast, the fry with Dh31 receptor knockdown in the chorizonin neuron consume the same amount of the protein containing food. So taken together, these results suggest that there is a separate population of the Dh31 receptor neuron in the brain that control feeding. We therefore perform another screen for the Dh31 receptor expressing neuron that regulate the protein feeding. So using Dh31 immunolabeling, Dh31 antibody immunolabeling, we found Dh31 receptor express another population of neuron in the brain. And beside the chorizonin neuron I mentioned before, another subset of the Dh31 receptor expressing neuron is aldostatin C positive. So we then hypothesize the circulatory Dh31 act on the Dh31 receptor alpha-studying C neuron to suppress protein feedings. So by knocking down Dh31 receptor in alpha-studying C neurons, the fry increase the total amount of amino acid in the feeding assays. And similarly, knocking down alpha-studying C in this neuron, fry also increase the intake amount of amino acid in feeding assay, suggesting that the Dh31 act on the brain alpha-studying C neuron to suppress protein feeding. And taken together, the gut-derived Dh31 act on two separate populations of the Dh31 receptor neuron in the brain to coordinate the switch from feeding to courtship. And one population release corresponding neuron to promote courtship. Another population releasing alpha-studying C to suppress protein feeding. So next, we further investigate whether there is a mutual interaction between these two neural pathways. And to address this question, we use a thermogenetic activation strategy to activate either corresponding neuron or alpha-studying C neuron receptively. So if there is a mutual interaction between these two neural pathways, we predict activation of the corresponding neuron would promote courtship behavior as well as suppress feeding behavior. And similarly, activation of alpha-studying C neuron would suppress protein feeding behavior and promote courtship behavior. However, thermogenetic activation of brain corresponding neuron in the male fry only increase their copulation percentage without any effect on their protein intake. And by contrast, thermogenetic activation of brain alpha-studying C neuron in the male fry suppress their protein intake and do not have any effect on their copulation percentage. So this result suggests these two neural pathways work independently to regulate feeding and courtship behavior, and corresponding neuron promote courtship, alpha-studying C neuron suppress feedings. And Dh31 from the gut act on these two independent parallel pathways to allow fry stop feeding and then transition to the courtship. And to conclude, our behavior result shows that a gut-derived hormone Dh31 respond to the amino acids and serve as a signaling molecule for the behavior transition from feeding to courtship. An ingestion of the amino acids trigger the release of the Dh31 by anterior endocrine cell from the gut. And finally, we found the circulating Dh31 reach the brain to activate two populations, two different parallel pathways to promote the male courtship behavior and suppress protein feeding behaviors. And this gut-to-brain communication provides a possible mechanism to accomplish a behavior transition between the physiological need and social needs. So as I showed you before, there are two hypotheses about how animals switch from one behavior to another. And one model proposed by Lawrence suggests that all drives compete and only one can emerge. However, my data shows that there is no mutual interaction between the corresponding neuron and alpha-studying C neuron pathways that drive feeding and courtship behavior respectively. And the other model proposed by Abraham Maslow is the different motivation drive established by the hierarchical combinator. And my data suggests that the Dh31 is a signal that indicates the low physiological need, such as feeding has been met, And in circuit level, I have shown Dh31 engage to different population of the neuron in the brain. One of them express cortisone, another express aldosterone. The cortisone neuron promote courtship and aldosterone suppresses protein feedings. And my result, the switch from feeding to courtship when the nutrient is adequate, supporting the model of the hierarchical organization in the different needs. And so now I have my own lab at SUNY Upstate, and we are in progress to investigate the underlying neural mechanism that regulate these nutrient-specific feedings. And as you know, the feeding behavior is important for the animal survival and health. And dysregulation of the feeding behavior can cause several feeding disorders, such as obesity and anorexia. And the feeding behavior is tightly regulated by energy homeostasis. However, having only energy homeostasis is not sufficient for the animal to maintain their physiological functions. You also need nutrient homeostasis. For example, the balanced intake of the specific nutrients such as amino acids and carbohydrate is essential to optimize the lifespan and reproductive success. The lack of the nutrient homeostasis can cause a negative impact of the fitness. However, there is ongoing debate whether the food intake is to maintain the energy homeostasis or to maintain the nutritional or homeostasis. So therefore, I want to investigate the circuit function of regulating such nutrient-specific feeding. And I hope my study of the neural circuit function in regulating this food intake in a simpler system can provide a new insight into the logic of the feeding regulation in general. So as I mentioned before, my recent study provide an entry point to understanding the neural circuit mechanism that regulate the protein-specific feeding. So we identify a protein satiety signal, Dh31, and Dh31 acts on the brain aldosterone neuron to suppress protein feeding. So with that, now there are many interesting questions we can start to investigate. For instance, how the Dh31 positive cell in the gut sends amino acid to mediate this Dh31 neuropeptide release, and how the aldosterone release from the Dh31 receptor neuron in the brain to mediate this protein-specific feeding behavior. And I think identifying such a neural architecture of the protein feeding will provide a new insight of wiring diagram logic that control the nutrient-specific feeding. And in the other hands, in general, the food typically contain multiple nutrients. For example, 2 major macronutrients, the protein and carbohydrates often coexist in the food in a various ratio. But it's less clear how the neural mechanism associated with the consumption of the different nutrients regulate the feeding behavior. So therefore, I want to investigate how the protein and sucrose signal are integrated in the brain circuit to regulate the feeding behavior. And identifying such a neural hub where the sucrose and protein signal integrate will provide an opportunity to understanding how the different nutritional signals are interact to regulate such feeding behavior. So with that, lastly, I'd like to thank the people who made this study possible. In particular, I want to thank my postdoc mentor, Jun Wen at UCSD for his support and his mentoring for my research project. And I also like to thank Dr. Qin Su for her help comments for my project. Additionally, I also like to thank the other members in our lab and the Dr. Qin Su's lab. And here is my funding source for this project. And thank you for your attention. I would like to take any question. And lastly, I want to share the advertisement for my lab. So we are currently in recruit a postdoc and research technician and PhD student as well. If you are interested in studying a nutrient-specific feeding regulation in a fly, and feel free to contact with me. And yeah,